Hello, my name is Professor Hudson, and you are about to view narrated PowerPoint lectures for Chapter 11 on genetics. In the last segment, we left off talking about the 7 characteristics of pea plants studied by Mendel. He studied flower color, flower position, seed color, seed shape, pod color, and stem length. Studying these individual characteristics one at a time allowed him to see patterns emerging. In the next segment now, we're going to talk about how single traits are inherited and how Mendel's work helped facilitate this understanding. So let's begin by looking at inheritance of flower color in pea plants. True breeding organisms possess traits that remain inherited unchanged by all offspring produced by self-fertilization. Mendel's cross-fertilization of pea plants used true breeding organisms. He cross-fertilized true breeding white flowers with true breeding purple flowers. The true breeding purple flowers only produce purple flowers, and the true breeding white flowers only produce white flowers. The parents used in this initial cross were known as the pea generation. The pea generation is the first generation that we are discussing in terms of the cross. The offspring of crossing this true breeding purple with a true breeding white flowered plant is considered the F1 generation. The F1 generation are the offspring of the parental generation. Note in this case, when he crossed a true breeding purple with a true breeding white flower, he obtained all purple flowers. On the surface, this suggests that the purple must be dominant over white. Now, to continue on with this cross, Mendel then took flowers in this F1 generation that were all purple and allowed them to self-fertilize. The F2 generation are the offspring of the F1 generation. What was interesting is that when he allowed the purple flowers to mate together, the F2 generation yielded purple flowers and white flowers at a ratio of 3:1, or 75% purple and 25% white. These results show that the white trait had not disappeared in the F1 generation, but was hidden as it came out again in the F2 generation. Mendel did crosses such as the one that was just discussed for lots of different traits. He looked at stem length, pod shape, seed color, flower position, flower color, and pod color. He employed numerical analysis in studying these traits. He followed the inheritance of these traits for several generations, counting the numbers of offspring with each type of trait. By analyzing these numbers, he saw that the basic patterns of inheritance were emerging. By counting the results of these crosses, he could figure out what was considered dominant and what was considered recessive. This is another slide just showing another version of the different traits that he looked at. Based on his work with these pea plants and these various traits, a 5-part hypothesis of inheritance of single traits emerged. First, each trait is determined by a pair of genes. Each organism has 2 alleles for each gene, one on each homologous chromosome. When 2 different alleles are present in an organism, the dominant allele may mask the recessive allele. Even though the recessive allele is still present, The pairs of alleles on homologous chromosomes separate or segregate from each other during meiosis. This is known as Mendel's law of segregation. Chance determines which alleles are going to be included in a given gamete or sex cell. Because homologous chromosomes separate during meiosis, the distribution of alleles to the gametes is random. True breeding organisms have 2 copies of the same allele for a given gene and are homozygous for that gene. Hybrid organisms have 2 different alleles for a given gene and are heterozygous for that gene. One of the key things that came out from Mendel's early work was the idea of Mendel's law of segregation. This states that 2 copies of a gene segregate from each other during the transmission from parent to offspring. This slide shows Mendel's law of segregation. For example, if we look at a particular gene called gene A, notice that during meiosis at metaphase 1 and metaphase 2, the pairs of alleles on these homologous chromosomes will separate or segregate from each other during meiosis, so that half of the chromosome material and half of the genes and alleles are found in each sperm or egg cell. Let's take a look at a YouTube video that further explains Mendel's law of segregation.
Mendel's second law of inheritance is now known as the law of segregation. During gamete formation, a diploid cell will split into 4 haploid cells. Remember, during meiosis 1, the homologous pairs separate. This also means that 2 different alleles coding for the same chromosome will separate. So let's try drawing this out. You start with a diploid cell, and we'll just say it has 2 chromosomes to simplify things. And during meiosis 1, the homologous pair separates, so each has only 1 chromosome, and then during meiosis 2, the sister chromatids separate, and as a result, you have 4 genetically different haploid cells. And so what this tells us, because each allele coding for the same trait is found on only one of the homologous pairs, that means when the homologous pairs separate, the alleles will separate with them. So for example, if you were talking about pea color, you have a yellow allele and a green allele. The yellow allele would be found on one homologous chromosome, and the green allele will be found on the other. And so when these homologous chromosomes separate into their separate gametes, one gamete will end up with the yellow allele, and the other one will end up with the green allele. So this is what the law of segregation states. 2 alleles coding for the same trait will separate during gamete formation. Mendel didn't know the specifics of genetics when he proposed this law. Pretty smart guy.
Let's also watch a brief YouTube video which has a song on segregation, which may help you to remember how it works.
Yeah, you know what it is. When I don't in my jeans, I do the thing. Yeah, but that's nothing. When I segregate my chromosomes, that's stunning. But in my genome, but my kids, they only get half. Segregation, 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 segregation. Half from mom, half from dad. You do the math. Segregation, 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 segregation. Gregor Mendez, genetic cross. Use P plus to see which other was the boss. Cross purebred, see which offspring they make. At one, all show the dominant trait. The phenotype is what he saw on the outside, but inside the genotype was heterozygous. Then he crossed the F1s with each other. He says the traits could be seen, but on a much lower level. From this we got Mendel's law of segregation. You get 2 alleles, 1 from each parent, but you only donate 1 to your own kids, cause they segregate in meiosis. Yeah. You know what it is. When I donate my genes, I do a thing. Yeah, diploid, that's nothing. When I segregate my chromosomes, that's stunning. Kept in my genome, but my kids, they only get half. Segregation, 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 segregation. Half in mom, half in dad. You do the math. Segregation, 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 segregation. Got a call from my teacher, this just in. Homozygous alleles, 2 that are the same. Recessively small, lowercase letters. Dominant or capital, they show through better. We got one of which we call it heterozygous, but the dominant allele is what the phenotype is. Got a genome of allele pairs that dictate the colors of my eyes, my skin, my hair. Yeah, you know what it is. When I donate my genes, I do the thing. Yeah, diploid, that's nothing. When I segregate my chromosomes, that's stunning. But in my genome, but my kids, they only get half. Segregation, 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 segregation. Half from mom, half from dad. You do the math. Segregation, 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 segregation.
So as you saw in the segregation mini lecture through YouTube and the segregation song, diploid organisms have two alleles for a given trait. However, when diploid organisms produce sperm and egg, the sperm and egg are always haploid or only have one version of the allele. This is due to Mendel's law of segregation. Let's take a look now at how exactly you can figure out inheritance of certain traits using something called a Punnett square. How are single traits inherited? Mendel's work led to the understanding of the origination of single traits of inheritance. Initial experiments included crossbreeding plants that differed in one trait. Punnett squares can be used to predict offspring genotypes and phenotypes from the combination of parental gametes. When doing a Punnett square, you first assign letters to the different alleles of the characteristic under consideration. Uppercase is used for dominant and lowercase is used for recessive. Determine the gametes and their fractional proportions from both parents. Write the gametes from each parent together with their fractional proportions in a grid and fill in the genotypes of each pair as if the sperm has fertilized the egg. From those, you can figure out the genotypic and phenotypic ratio, and you can figure out what the offspring may look like theoretically. Here's an example of a monohybrid cross with a Punnett square that was done in order to predict the number of tall and short plants that one might obtain when using a true breeding tall plant to fertilize a true breeding short plant. Notice in the F1 generation you get all tall plants. However, in the F2 generation you get tall and short plants. Tall is dominant and short is recessive. Here's a question that you can try as you watch the PowerPoint lecture. Attached earlobes versus unattached earlobes is governed by genetics. Unattached earlobes are dominant and attached earlobes are recessive. In the cross of a heterozygous for the unattached earlobes with another heterozygous that has unattached earlobes, the chances of having a child that also has unattached earlobes is, would it be 1 out of 2, 1 out of 4, or 3 out of 4? Why don't you try seeing if you can write out a Punnett square that would answer this question? And then the next segment of the narrated PowerPoint lectures for Mendelian Genetics, Module 7, Part 1, I will explain how you were to set up this problem. Let's pause here while you try to work this program or this problem on your own, and then I will explain it in the next segment.